You should be able to hear me. Good. All right, there were a lot of people who signed up, but it's only you and Rio, so I'm going to wait a few more minutes and then we can start. Oh, hang on, I see them right now. Okay, if you guys want, you can just type your questions in chat and I'll respond. I don't know why we only have two viewers. There's another one. Okay, I don't know if you guys can hear me. Can you guys hear me? No, the test isn't going to cover anything from chapter 10. No, just chapter 8, chapter 9. Yuma, for chapter 8, it's just mostly going to be enzyme regulation. So allosteric regulation, feedback inhibition. I mean, free energy, you'll need to know just because free energy is going to be used in the electron transport chain, but I mean, that's about it. I don't think you're going to see any graphs of exergonic versus endergonic reactions, but you definitely need to know how free energy is used by the cell ultimately to make ATP. Yeah, that's right, Jerry. Just a lot of chapter 9 and 8, and most of the questions from chapter 9 are going to be application-based questions. You'll have a few questions that are just pure knowledge, like vocab, but most of the questions will be application. I can sense your sarcasm, even in your typing. You don't have, the only intermediates that you might have to, or that you want to recognize are just the 4-carbon compound. You don't even have to know its name, but you just need to know that this 4-carbon compound is going to combine with acetyl-CoA. So these 2 carbons plus the 4 carbon equals the 6-carbon citrate. And beyond that, you don't have to know any of the names. You just have to know that as citrate is oxidized, it is going to lose carbon dioxide. And so technically new molecules are going to be created, but you just have to know them as intermediates. I don't know if you guys can see what I'm drawing on my screen. It still looks blank. What is the prosthetic part of the edited protein? I don't know, Yuma. Prosthetic part, that might just mean cofactors or coenzymes. So I think prosthetic part is just a general name for any non-amino acid any non-amino acid component that's helping a protein do its job. So like that heme group in hemoglobin is a non-amino acid component that's helping hemoglobin do its job. But you don't have to know all of the different names of the proteins that make up the electron transport chain. You just need to know that proteins do make up the electron transport chain, and those proteins are serving two main functions. The first one is to pass electrons, and then the second function is to capture the energy as the electron is passed and pump protons. The chain, I don't think ATP synthase is typically included as a component of the chain, because ATP synthase isn't really passing any electrons. It's just using the gradient that's produced from the electron transport chain in order to generate ATP. The chain, you can argue, ends with a protein, and then the non-protein component that makes up the end of the chain is just oxygen. Yeah, they might include it in some of the videos. I mean, typically it's included in all the videos, just because you really can't talk about the electron transport chain without talking about ATP synthase. I don't know, it technically might be considered part of the chain. That's not the way that I learned it, and I don't think that's the way that your textbook presents it either. You just need to know that it is a protein that's embedded in the inner membrane. It allows, or it does facilitate diffusion, so it allows hydrogen ions back into the matrix. And it's also an enzyme, so it's a big protein that has a couple different roles. One of the roles is facilitated diffusion, and the other role is using the energy that's released from that diffusion in order to generate ATP. So it's an enzyme that does a dehydration reaction and links inorganic phosphate to ADP. I'm presenting. I wasn't presenting before, so that might fix the drawing issue. I don't know. Hey, babe, I was planning on cleaning that. I was planning on cleaning that. Explain how protein structure is involved in enzyme specificity. So, Jared, tell me if you can see the drawing, but remember, enzyme specificity just means that the active site is highly selective for the substrate. So if you have an enzyme, and this is the active site, since all of the enzymes that we've talked about so far are proteins, and proteins have a very specific three-dimensional shape, it's important that these proteins fold properly so that the active site is able to interact with the substrate. So if a protein is misfolded, it can mean either the active site, there's a misfold in the active site, so maybe specific R groups can no longer interact with the substrate, or there might be a change in an entirely different region of the protein, but that might cause a ripple effect in the structure of the protein that would ultimately deform the active site. If you remember looking at hemoglobin and how hemoglobin binded to oxygen, that one single interaction caused a ripple effect across the entire protein that completely changed its shape. Chemiosmosis is just the process of hydrogen ions diffusing down their electrochemical gradient and being used to do energy coupling. And in this case, the energy coupling is creating ATP. ATP creation is an endergonic reaction, so it needs to be coupled with an energy releasing process. And the diffusion of solutes down their concentration gradient is an energy releasing process. Serious. It's the diffusion of hydrogen. It's the diffusion of hydrogen ions down their electrochemical gradient, so that as energy is released, the cell is capturing that energy and using it to do an endergonic reaction. And in this case, the endergonic reaction is ATP synthesis. I'm getting beeps. I don't know what the beeps mean. Is somebody's microphone on? I don't know if I'm missing questions or what.
The beeps mean that someone's like talking in the chat.
Oh, okay. I'm not seeing the chat scroll though. Oh well. Oh, I see. They're talking on the chat outside the call. Okay. Jared, cell fermentation, it occurs in the cytoplasm of a eukaryotic cell, and it also occurs in the cytoplasm of a prokaryotic cell, because there are no organelles in a prokaryotic cell, membrane-bound organelles in a prokaryotic cell. But even in eukaryotic cells, it occurs out here in the cytoplasm. And so this is one of the reasons why we think that glycolysis was probably one of the first metabolic strategies that evolved for living organisms, because it's found in both prokaryotes and in eukaryotes. And then even when you look at glycolysis in a eukaryote and you compare it to glycolysis in a prokaryote, virtually the same enzymes are used, the same intermediates are created. They're virtually identical chemical reactions. And sorry, fermentation also occurs in the cytoplasm. I thought you said glycolysis. It occurs in the cytoplasm. Yeah, I mean, Yuma, their job is to pump proteins, but then they're also passing the electron down the chain. If that electron passing job stops, then they can't pump anymore either, because they need energy from the falling electron to do their job, which is to pump. But yeah, after they create the gradient, the electron transport proteins, that chain isn't actually directly responsible for synthesizing ATP. What's directly responsible for synthesizing ATP is ATP synthase. Bella, the proton motor force, all that is just the force of, I don't know if you can see this, but it's the protons that have accumulated in the intermembrane space. This accumulation outside in the intermembrane space is creating a force that's going to be used to generate ATP. So the proton motor force and chemiosmosis are really similar definitions. It's just a force that's generated as the proton concentration begins accumulating and going up in the intermembrane space. Does it create the gradient? The electron transport chain creates the gradient, and the creation of that gradient produces the proton motor force. Evelyn, let me see. The point of fermentation, Evelyn, is to regenerate NADH. So remember, in glycolysis, you have glucose that is phosphorylated and then split into two molecules of G3P. And in order for glycolysis to become an energy producing phenomenon, in order for the cell to get ATP out of this, NAD plus must come in and oxidize G3P. When that happens, this G3P gets another phosphate, and that's going to happen to G3P on the top, and it's going to happen to the G3P on the bottom. So normally this NADH is going to go off to the electron transport chain if oxygen is present. If there's no oxygen present, it's not going to go to the electron transport chain, which means there needs to be some way to take this and turn it back into that. Because if the cell runs out of NAD+, then glycolysis can no longer continue happening. So the whole point of fermentation is just to ensure that glycolysis continues happening. Because if there's no oxygen, the cell can't do cell respiration. If it can't do glycolysis in addition to that, then it's in really big trouble because it has no other way of generating ATP. amino acid. Why would a cell need to generate more intermediates? The cell would generate, so basically those intermediates are just molecules that are created as the originals are broken down. So the amino acids are converted into intermediates of either glycolysis or the citric acid cycle. Those intermediates, the cell can do things with those intermediates. The cell can either choose to continue breaking down those intermediates and generate ATP, or it can take those intermediates and use them to synthesize a whole bunch of different organic molecules. So depending on what the cell needs to do, it will either continue breaking them down, or it will use those intermediates as like a template to synthesize a whole bunch of different organic molecules. Like, I don't know if you remember, but just as an example, when we were talking about human skin pigmentation, we looked at how the cell takes the amino acid tyrosine and it breaks it down into an intermediate, and then it uses that intermediate to build melanin. So it just depends on what the cell needs. Oh, I mean, there are thousands and thousands of different intermediates. I mean, just like you don't have to know the names of all the enzymes that partake in these chemical reactions, you're not expected to know the names of all of the different intermediates that could form as you break down these molecules. It's just way too much information for the scope of this class. Yeah, Jared, a redox reaction. Let me see if I can do this again. Maybe. A redox reaction, all a redox reaction is a transfer of electrons. So redox stands for reduction oxidation, and the easiest way to remember this is oil rig. So in a redox reaction, you're having atoms gain electrons, and you're having the atoms that gain electrons are gaining them because they're taking electrons away from something else. So redox reactions always happen together. There's always going to be a gain, and there's always going to be a loss. So one of the more simple examples is just sodium plus chlorine interacting to form sodium chloride. So sodium is going to lose an electron, chloride is going to gain an electron. So sodium has been oxidized, chloride has been reduced. So chloride is the oxidizing agent because it's causing something else to be oxidized. Sodium is the reducing agent because it's causing something to be reduced. I guess the more complicated example is like if you look at a combustion reaction. So if you take something like methane and add oxygen and you get carbon dioxide and water, the way that you tell what atom has been oxidized and which atom has been reduced is you just follow the hydrogen. So we start with carbon, and this carbon over here has lost hydrogen, and so it's lost electrons. Another way that you can think of it is that this carbon is now forming bonds with oxygen, which is a much more electronegative atom. So the oxygen is sort of stealing those electrons away from the carbon. So this carbon has been oxidized. This oxygen has been reduced because over here, first of all, the oxygen has gained hydrogens and electrons came with it. Also, look at this oxygen over here. it's in a non-polar covalent bond. Suddenly, it's now in a highly, highly polar covalent bond. So it's gained electrons in the sense that because of its electronegativity, it gets more electrons. So that's all a redox reaction is. And in chapter 9, we talk about them specifically because as electrons move, as electrons are transferred from higher potential energy to lower potential energy, in other words, as they move from reactants with high energy to products with lower energy, energy gets released. So for example, the movement of electrons from glucose to carbon dioxide and water releases about 680 or 678, or I don't know what I can't remember what the exact number is, but it releases a lot of energy, and that energy is captured by the cell in order to ultimately generate ATP. Evelyn, are the intermediates similar? No, the byproducts are different from the intermediates. Evelyn, the intermediates are just the organic molecules that are formed as your food is being broken down. The byproducts in this case are water. The byproducts of cellular respiration are water and carbon dioxide. The intermediates are like, if you look in your book and you look at the figure for citric acid, for example, it starts with that 6-carbon molecule and then it goes through the cycle. And I think your textbook names all of the different molecules that are formed, all of the different organic molecules that this citric acid turns into as it's oxidized. And as those intermediates form, carbon dioxide is given off as a byproduct. Jared, in the book, it talks about Ellie, they interact with oxygen to form, that's fine, Yuma, they interact with oxygen to form water, and that water can be used for hydrolysis. Some of it is breathed out, but yeah, those hydrogen ions need to be replaced, and they're replaced by the food that you eat, because that's where those hydrogens originally come from. They're stripped away, along with electrons, they're stripped away from food. Okay. That's right, Jared. Is there a question there? Are you helping somebody else with something? Again, the ATP is created. The ATP is created not by interacting with electrons, but if this is your inner membrane, and these are the electron transport proteins, okay, so you have NADH dropping off the hydrogen and electrons, you have FADH2 dropping off hydrogens and electrons, and as those electrons fall through the chain, these proteins are going to pump hydrogen out into the intermembrane space. That stored energy is going to be used to create ATP. So over here you have ATP synthase. The hydrogen ions diffuse through ATP synthase. And as they do that, energy is released, and that energy is captured by ATP synthase to take ADP and inorganic phosphate and do a dehydration reaction to make ATP. So notice that the electron is over here. Whoops, it's over here. The electron itself isn't directly interacting with these reactants. That electron is eventually going to get captured by oxygen, which is going to, as it captures that electron, that oxygen is also going to interact with these hydrogen ions that are diffusing through ATP synthase to make water. The point of the electron is to generate stored energy in the form of a proton gradient. These high energy molecules are unstable, and they're not good for storing energy. The concentration gradient that's created is much better for storing energy. It's sort of like a little cell battery. So it's much better for storing energy, and the mitochondrion can tap into that energy when it needs to in order to create ATP. The citric acid cycle happens twice because for every 1 glucose you start with, you end up with 2-pyruvate. So if you end up with 2-pyruvate, each pyruvate is going to get converted into an acetyl-CoA. So you have 2 acetyl-CoA. Each acetyl-CoA is going to interact with that 4-carbon molecule called oxaloacetate to form citric acid. So for every 1 glucose you start with, you get 2 citric acid cycles. Yeah, so it's the latter part of your question. It's the fact that there are 2 acetyl-CoA. Okay, again, glycolysis creates ATP. You have 1 glucose. That glucose is phosphorylated twice by using 2 ATP molecules. That makes it unstable, and it makes it so that glucose splits in half like that to form 2 molecules of G3P. Each molecule of G3P has 1 phosphate group attached to it. So far, the cell isn't getting any ATP back. In order to get any ATP out of this, NAD+ is going to oxidize G3P, and NAD+ is going to be reduced and turn into NADH. That releases energy, and that energy is used to attach an inorganic phosphate, meaning this phosphate was free floating in the cell cytosol or in the cytoplasm. That's going to happen down here too. Now we have 4 phosphates, which means that the cell can rip each phosphate off and transfer it onto an ADP to make ATP. So this is called substrate level phosphorylation, because we're using an enzyme to rip these phosphates off and transfer them directly onto ADP. So that's how glycolysis creates ATP. Concept 9.6. Yuma, you just need to know basically what we did with Patrick. We saw that figure where different food molecules plug in different pathways. So you just need to know, for example, that amino acids can plug into glycolysis or the citric acid cycle. Fatty acids can plug in directly into acetyl-CoA. And that's important because we use that knowledge to either diagnose metabolic disorders or even treat metabolic disorders. So like with Patrick, he couldn't convert pyruvate into acetyl-CoA, so we fed him a high-fat diet because fatty acids can bypass pyruvate and turn directly into acetyl-CoA. I'm pretty sure that we can have more than 10 people in at the same time, because right now there are 14. I just don't think people understand how to join the chat. Yes, Jared, phosphorylation is adding a phosphate to something. And it's, I mean, it's pretty important when you use it to, like when you phosphorylate ADP to make ATP, that's pretty important. In this chapter, yes. But in chapter 11, we're going to talk about different types of phosphorylation that are also extremely important for cell survival. Different amino acids enter in different steps. And I don't even know which ones enter which step. I just know that some enter into glycolysis, some enter into and are converted into intermediates of the citric acid cycle. But I don't know which ones go where. Okay, Patrick guy, yeah. Let me see. So here was the problem with Patrick. Patrick could take glucose. He could convert it into pyruvate, so his glycolysis was fine. There was nothing wrong with any of the enzymes that perform glycolysis. His disorder was the pyruvate was unable to be converted into acetyl-CoA. And if you remember, acetyl-CoA is a bridge that links glycolysis to the citric acid cycle. So you have to go in this, if you're talking about glucose, you have to go in this linear pathway. But for Patrick and for anyone, if you, instead of consuming glucose, you consume fatty acids, those hydrocarbon tails that are so common in fats, those fatty acids are thanks to a different set of enzymes, a completely different set of enzymes. So remember, pyruvate goes to acetyl-CoA thanks to this enzyme, this massive enzyme called pyruvate dehydrogenase complex. Different enzymes turn fatty acids into acetyl-CoA. So what they tried to do with Patrick in order to help him generate ATP is they fed him a high fatty acid diet. Unfortunately, though, for Patrick, is the brain or the central nervous system really relies on glucose in order to get ATP. So feeding him fatty acids is going to help him for some tissues, but it's really not going to help him with his central nervous system. Rio, what makes fatty acid acetyl-CoA? It's a process called beta oxidation, but I don't know the names of all the enzymes. I haven't committed them to memory, and you don't have to know the names of the enzymes either. You just need to know that a different set of enzymes turns fatty acids into acetyl-CoA by oxidizing those fatty acids. Yuma, let's see. And okay, let me see if I can find a figure and then screen share that figure. So give me a second. Okay, Yuma, are you able to see this figure with all of these molecules? Okay, so this is what I mean by intermediates. We have glucose, we have glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, like all of the names of these organic molecules that are created as glucose is oxidized and rearranged. So there are enzymes that transfer there are enzymes that transfer phosphate on There are enzymes that rearrange the covalent bonds and create isomers. And remember, isomers have different names because they are molecules that are connected differently. So fructose and glucose, for example, are different molecules because they have different shapes, because their covalent bonds are connected in different ways. So an intermediate, by definition, is all of these organic molecules that are formed as the enzyme works on glucose. So all these names in black right here are intermediates, and I'll pull up another figure of the citric acid cycle intermediates. One second, I'm responding to an email. Okay. Let's do this one. No, that's not. Here we go. Okay, so citrate, isocitrate, ketoglutarate, all of these names right here in these colored boxes, those are intermediates. And again, all you have to know is here's citrate, which is that 6-carbon molecule, and then here's the 4-carbon molecule that interacts with acetyl-CoA right here. All of the names of the enzymes that are in red and all of the names of the intermediates, you don't have to commit to memory. The intermediates are just important because they are critical to the oxidation of citrate. Like if we can't make isocitrate, for example, we can't continue oxidizing. So they're critical for the complete oxidation of glucose or citrate. And they're also critical because they can be pulled out of the cycle by the cell, and they can be used to make other compounds. So if the cell doesn't need them for energy, it can create those intermediates so that it can then build different molecules. Yen, that's the standard error of the mean. You just need to know what we went over today. I mean, we talked about how the standard error of the mean is used to look at whether or not the average in two different groups is different, or whether those differences aren't real and due to chance or another variable that we're not accounting for. Jared, you just need to know that all of the food that you eat can plug into different aspects of glycolysis or cell respiration. but you don't have to know specifics because they cannot see the chat box. Yeah, I can read out loud the questions so they can see what they can hear me and they can see what I'm writing, but they can't see the chat box. Okay, yeah, I can restate the questions. Oh, okay. So they're probably watching this on my YouTube channel, but they're not actually in the Hangout. That makes more sense now. Let me see if I can. Can anyone post the link in the chat to the streaming video so that I can send it to people who can't join? Thank you. Oh, thanks, Ria. I missed it earlier. Sorry. Yes, I'll post it on my website right now. Okay, so I'll try to go for like 30 more minutes, and then we'll call it a night. And remember, I'll be here at 7:30 in the morning to do more review. So Hume is asking about the standard error of the mean, and he's asking why I multiplied the standard error by 2. So the answer to that question is, let me pull up the notepad. The answer to that question is, by definition, 2 standard errors of the mean gives us a 95% confidence interval. So in other words, if we only calculated 1 standard error of the mean and then we added that to the sample mean and then we subtracted that from the sample mean, that would give us a certain interval, and we would be 68% confident that the true population mean falls within that interval. So if we want to be more confident, we have to increase our interval. We have to I mean, I don't know how else to say it. We have to increase our interval. So if this is the sample mean, and we wanted to do our 1 standard error of the mean, and we wanted to be 68% confident, that's really not confident enough in the scientific realm. So if we want to be more confident, we have to be willing to accept a larger spread. And then I could be 99%, if I wanted to, I could be like 99% confident, but in order to be 99% confident, I have to be willing to accept an even larger spread. So in the scientific community, the threshold is 95% confidence, and in order to get 95% confidence, you have to use 2 standard errors. So in order to calculate 1 standard error, all you have to do is you have to take the standard deviation of the sample and divide it by the square root of the sample size. And then whatever number you get, you just multiply it by 2, and then add that to the mean and subtract it from the mean. By 95% confident, what we mean is that since we're calculating the average based on a sample, we really have no way of knowing what the actual true population average is. The logistics of sampling an entire population are a nightmare, and it's super expensive. So as researchers, what we do is we take samples, we take smaller groups, and we try to use the sample statistics to get an idea of what the true population looks like. So since we're not actually measuring the entire population, all we can do is we can get a good idea of what the population looks like. And so in this case, that's what the 95% confidence means. It means that we don't know for certainty what the actual population statistic is, but we can be 95% confident that it's somewhere between these numbers. These numbers simply being the sample mean plus 2 standard errors, and then the sample mean minus 2 standard errors. And so that's our confidence interval. Where do the products? So Ellie's asking, where do the products of one turn of the Krebs cycle go to afterwards? The products of one turn of the Krebs cycle. So there are a couple of different products of the Krebs cycle, or the citric acid cycle. So you've got citric acid being oxidized and basically getting converted back into the 4-carbon molecule called oxaloacetate, so that the cycle can start all over again. So you could argue that one of the products of the citric acid cycle is its purpose is just to recycle oxaloacetate so that the cycle can keep going as long as we're bringing in more acetyl-CoA. The other products of this cycle, like you said, are FADH2 and NADH. You also have some ATP getting made, which is going to be used by the cell to do work. But yeah, these 2 products are going to go to the electron transport chain and deposit their hydrogens and their electrons into the chain so that proton pumping can happen. Let's see. Yeah, so like when you say these leave it to go to the last step, yeah, they leave the matrix, they go to the mitochondrial inner membrane, and when they get there, they're stripped of those electrons, and those electrons are fed into the chain, and they turn back into NAD plus and FAD, and now they're able to go and get reduced again. So they go back and do more oxidation, get more electrons. Someone's asking Chapter 8, what information from Chapter 8 is going to be on the exam. Most of the information from Chapter 8 is going to cover allosteric regulation, feedback. There was a really good chart in Chapter 8 that basically it was like A forms B, forms C, forms D, but then A could also form F or something like that. So all of these arrows represent enzymes, and if an enzyme is non-functional, which was exactly what was wrong with Patrick, instead of A turning into B, the only thing it can do now is turn into F. So for Patrick, A would be what, pyruvate, and instead of pyruvate being converted into acetyl-CoA, for Patrick, the only thing left for it to do was get converted into lactic acid. So yeah, that's the biggest part of Chapter 8 is enzyme regulation. Yuma is asking, calculating values when graphing the data. If anything, I'll see if I can. Never mind. No, you're not going to have to count. You're not going to actually have to calculate the standard deviation. You might have to calculate the average. But the only questions that I've seen on AP exams that involve the standard error of the mean, they actually tell you what the standard error of the mean is. And so at that point, all you have to do is just add it to the mean and subtract it from the mean, and then plot it on a bar graph and look at whether or not those error bars overlap. Yeah, you'll need to do it twice. If they give you one standard error, you're always gonna multiply that standard error by 2 to get the 95% confidence. If you only do just one standard error, you're not 95% confident, you're only 68% confident, and that's not confident enough in the scientific community, right? So you can say with 95% confidence. Okay, I have a student asking me to explain the difference between substrate level phosphorylation and oxidative phosphorylation. So substrate level phosphorylation is going to happen in glycolysis, and it's going to happen in the citric acid cycle. Oxidative phosphorylation is happening at or thanks to the electron transport chain, and the enzyme that's actually doing it is ATP synthase. So what's happening in substrate level phosphorylation is you have organic molecules that have been phosphorylated. So those phosphate functional groups either came from ATP, or they could have also come from free-floating phosphate that was attached to the molecule as it was being oxidized. But in substrate level phosphorylation, all that's happening is an enzyme is, like, here's the active site. Here's an enzyme. An enzyme is binding to the molecule with the phosphates. It's also binding to ADP, which has 2 phosphates instead of 3, and it's ripping the phosphate off of this molecule, for example, and it's transferring it directly onto ADP. And so ADP is going to get that phosphate and turn into ATP. So that's substrate level phosphorylation. And the difference is that in oxidative phosphorylation, what's happening is you have the mitochondrial inner membrane. So here's the matrix. Here is the intermembrane space. In oxidative phosphorylation, you have the electron transport chain pumping hydrogens out. And that electron is caught by oxygen as it reaches the end of the chain. That's why it's called oxidative phosphorylation. But those hydrogen ions are going to diffuse through ATP synthase. And ATP synthase is going to use the energy that's released as these ions diffuse into the matrix. And it's going to take ADP and free-floating phosphate and attach that free-floating phosphate onto ADP to make ATP. So the difference is over here, we're ripping phosphate off of something, like we're stealing it from one molecule and giving it to ADP to make ATP. Over here, we're using the flow of hydrogens to allow this enzyme called ATP synthase to take free-floating phosphate and put it directly onto ADP. And this process is going to result in much more ATP than substrate-level phosphorylation. Every textbook has a different number, but it ranges from between 32 to 34 ATP per glucose molecule, as opposed to substrate-level phosphorylation, which is something like you only get 4 ATP per glucose molecule. So this is a much more efficient way of generating ATP. Yeah, I mean, you need to know, I had a student ask how much you need to know about fermentation. I think I have drawn something. Yeah, so here's what I have drawn about fermentation. So you just need to know that, number one, fermentation happens in the cytoplasm. It happens when there is no oxygen present. And the reason it happens when no oxygen is present is because this NADH that gets made during glycolysis, you see down here, remember that when the NAD plus oxidizes G3P, that energy that's released is used to add a free-floating phosphate onto G3P. If that process doesn't happen, the cell cannot get 4 ATP out of glycolysis. So if this NADH can't go to the electron transport chain and turn back into NAD+, the cell must have a way to turn this NADH back into NAD+. Well, if it can't use the electron transport chain because there's no oxygen, it's got to use something. And what it uses is pyruvate. So NADH is going to dump its hydrogen and electrons onto pyruvate. That pyruvate is then going to turn either into lactic acid or, depending on what organism we're talking about, ethanol and carbon dioxide. Right? So these are byproducts that are created, and the purpose is to turn this into that. Because if the cell runs out of NAD+, then glycolysis cannot produce ATP. And if the cell isn't getting ATP from respiration, and it's not getting ATP from glycolysis, it's in a lot of trouble. Yen, you just, a student is asking about the enzyme lab. You just basically need to know that enzymes are, because they're made up of proteins, they are sensitive to environmental change. So that could be temperature changes, changes in pH, and if we change the environment, we could possibly denature the enzyme. If we heat up the solution, we could increase the rate of the reaction because we're increasing the frequency of collisions. But if we heat it up too much, we can actually start breaking the hydrogen bonds that hold the enzyme together. And as soon as we do that, the enzyme becomes useless. So high temperature can speed it up. Too high of a temperature can actually shut the reaction down by making all of the enzymes unfold. And then, of course, low temperatures are going to slow down the rate of a reaction because slow temperatures mean that the molecules are moving more slowly and colliding less frequently. And then as far as how pH affects enzyme form, remember that pH is just a measurement of hydrogen ion and hydroxide ion concentrations. So since these are charged particles, they can interfere with hydrogen bonding and they can interfere with ionic bonding. They can also interfere with R groups in the active site of an enzyme. So if we start messing with pH concentration, we can begin interfering with the bonds that stabilize a protein's three-dimensional shape. And so, I don't know, you might, or I expect you to know if on my x-axis I have various temperatures and on my y-axis I have the initial rate. Remember, when we're calculating or when we're trying to determine enzyme function or enzyme activity, we're always going to measure the initial rate of a chemical reaction in the presence of an enzyme. And so let's say I conduct, I don't know, 6 different experiments. And I get something like this. Right? So this tells me that, number 1, this point is the enzyme's optimal temperature. This is the temperature where the enzyme functions best or works the fastest. And then if I go to the left and start lowering the temperature, that's going to slow down the rate because the enzyme and substrates are going to move more slowly. And then once I start getting too high of a temperature, I'm going to interfere with the bonds of the enzyme until eventually the temperature is so high that the hydrogen bonds completely break down and the enzyme completely unfolds and there is no more active site. Which was basically all you guys did in the lab. You guys messed with temperature, you messed with pH. Some people messed with some other stuff, but that was the gist of the lab. All right, about 10 more minutes and then I'm going to hit it. What I'll do in the future is I'll have a form where students can submit questions because it looks like the only way you guys are able to submit questions is if you're in the chat. Or no, I guess you can type your questions out if you're I don't know, if you're on the web page that you go to when you click on the invite. Yeah, so it says that there are like 32 people in the conversation. Feel free to type questions into that chat log, even if you're not in the actual Hangout or not in the video feed. Yuma's asking about the Spit Lab. The Spit Lab was just a demo. I mean, it was basically the exact same thing as you guys had done. It's just that instead of looking at peroxide or peroxidase and hydrogen peroxide, we were doing amylase and starch. The concept is the exact same. And then you're also asking about the virtual lab. I mean, I expect you to basically be able to tell me how we measure the rate of respiration. And the best way to do it with the equipment that we have is just to look at oxygen consumption. And the way that you do that is you look at, or the way that you have to do that is you have to use potassium hydroxide to take carbon dioxide out of the microrespirometer so that you actually can see a decrease in the pressure of the system. And that decrease in the pressure of the system is going to cause water to move. So if this is your microrespirometer, you have a little capillary tube, you have a syringe, and there's the plunger. You have your potassium hydroxide down here. You have some cotton right here so that the organism doesn't come into contact with it. And then you just have your organism. In this case, it's germinating peas. And as these peas germinate, the pressure in the chamber goes down. And since these microrespirometers are underwater, as the pressure in the chamber drops, water actually gets pushed further and further down into the tube. And so we can measure the distance that the water has traveled, and that gives us an idea about, or it allows us to quantify, it allows us to put into numbers how rapid these organisms are respiring. And so, like, I expect you to know how endotherms might consume oxygen at different rates compared to ectotherms, or why a germinating pea might have greater oxygen consumption than a non-germinating pea, or why, if temperatures drop, a mammal might have higher oxygen consumption. So those are the big points from the virtual lab. I have a student saying CO2 was building up. Yeah, so the whole problem here is normally when cell respiration happens, oxygen is consumed by the organism, and that oxygen is replaced with carbon dioxide. So over here, if you're trying to measure oxygen consumption by looking at pressure changes, there is no pressure change if you don't do anything with this carbon dioxide, because as soon as you take oxygen out, you replace it with carbon dioxide, so the pressure of the system doesn't change. So that's why we have potassium hydroxide, to help eliminate the carbon dioxide so that there is no carbon dioxide replacing oxygen. You just have oxygen consumption. And that oxygen consumption is what's causing a decrease in the pressure inside the chamber. And that's what's causing the water to get pushed down into the microrespirometer. Oh God, I think I just explained that with my microphone muted. Okay, never mind. You're welcome. So I'll maybe hang around for another minute and see if there are any more questions. But if not, I'm going to go ahead and end the call or the Hangout. You're welcome. All right, that's it for tonight. I'll be holding the review session tomorrow starting at 7:30 and probably end it at around 8:10 or 8:15. So you guys can stop by whenever you want. You can stay the whole time. You can leave early. You can come in late. But yeah, all right, I'm out.